All right, well, it's 12:01, and why don't we get started? I want to thank everyone for joining our restart of Cancer Center Grand Rounds. We put a pause on Cancer Center Grand Rounds about a month ago, and I just want to say that what's happened in the past month among so many individuals in our cancer center to respond to this pandemic is nothing short of heroic. From redeploying our clinical services, moving out of Smilow Cancer Hospital, finding new ways to care for our patients and ensuring that their cancer care is moving forward. On our research side, our clinical trials organization has reorganized to continue to provide the ongoing care needs for patients on clinical trials through various technologies and maintaining their safety. In terms of our training programs and with respect to bringing our trainees into this process, but at the same time assuring their safety. And I think equally important is our basic research laboratories, who have obviously had to suffer a bit of a pause during this period, but at the same time have thought of new and innovative ways to address this. I think one very important example, many of you may know, was that the National Cancer Institute about 10 days ago issued a request for applications for supplements for cancer center members who were working on COVID. 30 individuals in the cancer center submitted applications in a 48-hour period. We were able to submit those applications to the NCI, which continue to be under review. But I think it's just a reflection of all the extraordinary work that our center is doing. And I think today's speaker is emblematic of how individuals have stepped up to address this crisis. We're very fortunate in terms of our restart of Cancer Center Grand Rounds to have as our speaker, Dr. Akiko Iwasaki. And Akiko, as you know, is the von Zedwitz Professor of Immunobiology, Molecular, Cellular, and Developmental Biology. She is a Howard Hughes Medical Institute investigator. She is a valued member of the Faculty of Immunobiology, as well as our Cancer Immunology program in the Cancer Center. And her work over the years has been sentinel of understanding of the immunologic responses to viruses and adaptive immunity, and more recently work in cancer immunology. In response to this latest crisis, she and her laboratory have stepped up work to improve our understanding of the immunology of COVID-19. For all of her work, Akiko has received numerous honors, most recently election to the National Academy of Science in 2018 and the National Academy of Medicine in 2019. So Akiko, thank you very much for helping us restart this. We have a lot of folks online, and we welcome hearing your work.
Thank you, Dr. Fuchs. Can you hear me?
Yes.
Okay, great. It's my privilege to be able to present this work today as a restart of the Cancer Center Grand Rounds. And today I was going to focus on our response to COVID-19, in particular, some of the work that I'm doing around both the surveillance and basic research that we are doing as a laboratory to deal with this pandemic. And so a lot of what I'm going to tell you today is very preliminary. We just literally began 10 days ago, some of the research that I will share with you, but I thought it would be informative, particularly many of you dealing with cancer patients, to be able to kind of get a glimpse at what we're doing and possible collaborations, hopefully with your group to deal with this pandemic. So this is a first slide that kind of looks at my personal activities around COVID-19. And this will also kind of give you a sense about what else is going on in Yale around this virus. So I serve on the COVID-19 Laboratory Task Force, which is led by Dr. Albert Ko and Dr. Saad Omer, as well as many members that are critical to this team is Nathan Grubel, Ellen Foxman, and Marie Laundrie. And we began this effort, in the very beginning of March. And we are rapidly ramping up our effort to be able to try to provide COVID-19 testing in the Yale New Haven Hospital, as well as in the healthcare community, healthcare care worker community at Yale. And I'm also working with the HIPC to provide some repositories that will become available nationwide with Dr. Ruth Montgomery and David Hafler and Al Shaw. And this is an important work, which is a part of a national effort to collect tissue samples and analyze them in high throughput manner so that we understand the immune response to this virus much better. I'm also coordinating and collaborating with multiple investigators on various aspects of immune response to this virus, such as serology assay with Aaron Ring, neutralization assay with Craig Weiland. I'm also part of a team which is looking at a home test kit that's being developed by 4Catalyzer, which is a startup biotech company out of Guilford. So this is would be a great thing to have in order to be able to test COVID-19 RNA status in a home test kit, and that can be widely available to people. And we're also collaborating with Eric Meffrey to look at generation of protective antibodies and B-cell receptor repertoire analysis with Steve Kleinstein. And there are many other collaborations that I didn't list here, but this is just to point out that this is a wonderful community of collaborative individuals that are at Yale trying to really deal with this virus in a timely manner. And I'm fortunate to be around people who are like-minded and really service-oriented. And I'm also trying to help with the acquisition of PPE and swabs and tubes, all the supplies that are necessary to work with the biorepository, which I'm going to come to next in a few slides. And then I'm also doing personal communication about the science behind this virus on various media and social media. So my laboratory has been working around the clock to try to help out in various activities, such as performing the PCR COVID-19 test for inpatient, outpatient, and healthcare workers in close collaboration with doctors Charles De La Cruz, Shelley Farhadian, Nathan Grubal, Albert Coe, and many others. I'm sorry, my dog is barking. We are also conducting isolation of PBMC and plasma for the biorepository and training people to become BSL-3 certified to work with infectious COVID-19, SARS-CoV-2. And I'm going to spend a little time talking about my lab's research activities on COVID-19. This involves a multi-pronged approach. One is the creation of a mouse model of COVID-19. So the receptor for this virus, the human ACE2, is expressed, the human version of it is a receptor for the SARS-CoV-2, but the mouse ACE2 is not a receptor. So what we're trying to do is to make a AAV or lentivirus vector induced expression of this receptor in the lung and other tissues to be able to study the spread and the disease of this virus in a mouse model. And most recently, we are also collaborating with Richard Flavel's lab, who has created a very advanced model of humanized mice to be able to study human immune response in the setting of the mouse model. We will investigate both innate and adaptive response to this virus and what confers protection versus what is pathogenic. We're also doing some analysis, development of mouse-adapted SARS-CoV-2, which is also useful because this can be rapidly applied to any knockout or transgenic mouse models that we have by adapting the human SARS-CoV to replicate in the mouse. And that will also, obviously, anything that we do will be widely available to the community and, to be able to study this pathogen. And then analysis of the PBMC from COVID-19 patients. This is a large collaborative effort that my lab is participating in, and I'll show you some data on that today. We are also trying to develop a better multiplex PCR analysis for COVID-19. This would obviously be very helpful in terms of trying to reduce the number of wells that are required to test for COVID-19. Currently, we're using multiple wells per person, per sample, in order to do PCR. And this is one of the things that my lab is working on, and examination of various antivirals against COVID-2. So for the creation of mouse model of COVID-19, one of the ideas is to just sort of give a wild type animal or any animal, knockout, whatever we want, AAV encoding human ACE2. And this can be delivered intranasally to have respiratory expression of the viral entry receptor. And then we can infect such mice with COVID-2 to be able to study infection and disease, and of course, immune response. And we are also collaborating, as I mentioned before, with Nick Joshi's lab to do a parallel approach using lentivirus to express the ACE2 receptor, and also with Richard using his Mr. G6 mice, which is I guess the latest update on his humanized mouse model, to be able to study infection in mice. The other approach, as I mentioned, is the mouse adaptation of this virus. So we will use, again, multi-pronged approach to get to this. One is to infect the mice multiple times repeatedly with the human SARS coronavirus 2. We'll isolate virus from the respiratory tract after a few days of infection, and repeated passaging of such virus in the mice will allow us to hopefully grow out variants that would be capable of replicating in mouse tissues. And this will circumvent any need to express the human ACE2 in future animal research. The other possibility that we're other approach that we're taking is to passage the virus in vitro and then give it to the mouse in order to kind of speed up the process of this adaptation. And so the in vitro adaptation is currently going on in Dr. Weiland's laboratory, and also molecular adaptation is being taken on by Dr. Brett Lindenbach's lab, where he can introduce specific mutations that are known to cause adaptation of SARS-CoV-1 virus and introduce that into the COVID-2. So hopefully, with all these multiple approaches that we and the collaborators are taking, we can start to have a reliable mouse model to study COVID-19 infection and immunology. And I'm going to take a couple of minutes to describe the respiratory infection biorepository. The PI of this repository is Dr. Albert Koh from the Yale School of Public Health, and he is heading this effort to be able to collect various samples, such as nasopharyngeal, oropharyngeal, as well as blood, saliva, rectal swab, urine. You can see all these samples that are being collected from patients who consent to enrolling in this biorepository. And so there are 3 cohorts of patients. One is the inpatient cohort, and the other is the outpatient cohort, as well as the healthcare worker cohort. And each one of these cohorts provide a unique set of insights. For instance, in very severe diseases in ICU, how are the patient's immune response being generated, and what are correlating with protective versus those recover versus those that go on to progress in disease, as well as the healthcare workers is a great cohort, because most of them are obviously not infected when they enroll, and if there's any exposure, we can tell right away from the NP swab whether there's any PCR positive viral RNA, and then we can inform those volunteers about their status of the COVID-19, as well as to study their immune response. So this is an ongoing biorepository, and so many people are involved in this. And so today I thought I would tell you a very preliminary data on the PBMC analysis from COVID-19 patients that my laboratory is participating in. And this is a work of very hardworking individuals in my laboratory, four of whom are listed here: Carolina Lucas, John Klein, Julio Silva, and Patrick Wong, and many others that are involved. So what we do is we, from the biorepository, we collect the PBMC patients. And today, mostly what I'm going to tell you about are the inpatient cohort, but we're expanding this to the outpatient as well as the healthcare workers. So today's talk is mostly focused on the inpatients. And so samples received from very recently, March 23rd, we started analyzing. And so what we do is we use flow cytometry to analyze various lymphocyte subsets and myeloid cell subsets, and kind of focusing more on the T cell activation status, memory subsets, and also functional readouts such as cytokines and granzyme from these T cell subsets. So the purpose of me sharing these data it's not a conclusive data at all. It's sort of a very early glimpse of what we're seeing. And we hope to really increase the number of patients analyzed, as well as looking at viral load in each patient. And thanks to the biorepository, we have access to all of this information. But I just thought I would share this very preliminary glimpse today. So this is looking at just overall PBMC composition and divided by patients. And you can see that there is variability between the composition in some patients. Some patients have accumulate a lot of neutrophils, and others are predominantly T cells and B cells. Anyway, this isn't divided in terms of severity of infection or anything like this, just an overall glimpse. And this is important because to note that the biorepository allows us to look at a time course of each patient. Every 3 days, these patients' PBMCs are collected. And so we can look at a time course of how these patients' immune response corresponds to their clinical outcome and a viral load. So in very few patients that we've looked at so far, we see that the patients that are in red, we are basically depicting those that still remains hospitalized, and those that are blue are discharged patients. So there are some statistical significance with respect to the white blood cell count in the first look at these blood when we first enroll these patients into the study between hospitalized and discharged groups. And then lymphocyte count is variable. It's known in the literature that lymphopenia is one of the things that happen in severe cases of COVID-19. And this, because we're only looking at one snapshot at the very first blood draw from these patients, at this point, we don't see a lot of difference yet. Monocyte counts, again, there seems to be some correlation between those that are hospitalized versus discharged in terms of the monocyte count. And this is also consistent with the literature. Looking at cytokine production from CD8s and CD4 T cells, what we see is some, this is just overall snapshot of what patients' T cells are making with respect to cytokines. And you can see that some of these patients make multiple, T cells make multiple cytokines, whereas others, other T cells are making no cytokines at all. And again, there is no meaning to this other than just to show you that we are measuring these cytokines from these T cells. Oops. I don't know. Sorry. Okay. Somehow I cannot proceed. Oh, here we go. And this is looking at individual data with respect to hospitalized versus discharged patients' PBMC T cell response. Again, I don't want to make any conclusions at this point, but we do see variability in terms of interferon gamma secreting cells and other, for instance, the TNF secreting cells between hospitalized and discharged patients. But none of these are right now significant. We're just sort of collecting more and more data to be able to see if there's any difference that's significant. And drawing some attention to a couple of patients for which we do have 2 time points. So patient number 1, indicated in this purple color, has an ongoing disease, whereas patient number 6 was discharged. And just looking at the progression of disease with the patient number 1, we see an accumulation of the exhausted phenotype of T cells in CD4s and CD8s, whereas patient 2 seems to just have similar percentages of these exhausted T cells within that person's peripheral blood. So again, this is a very preliminary look, and we're expanding these numbers now. And looking at the other functional features of these T cells, we see that CXCR3 positive CD8 T cells, which are capable of migrating to infected tissues and performing their effector functions, seem to be a little lower in the hospitalized group, whereas interferon gamma secreting, TNF secreting Th1 type of cells may be even enriched in the hospitalized group. There are a couple of other interesting myeloid features as well. We're seeing that monocyte count tend to be lower in this is classical monocyte, not the inflammatory monocytes, tend to be lower in the patients that sort of are hospitalized versus discharged. And similarly, the dendritic cell subsets that we're looking at, the classical dendritic cells, appear to have lower percentage in the hospitalized group. The other features that we see from the cytokines analyzed from these patients is that IL-6 is elevated in patients with progressive disease. And this has been published by a couple of groups already, that blood IL-6 levels, higher levels of blood IL-6 correlate with worse prognosis. And then similarly, soluble IL-2 receptor is elevated in more severe cases compared to those that go on to get discharged. So this is interesting because IL-2 receptor elevation is seen in other diseases like hemophagocytic lymphohistiocytosis, and kind of really indicating that this disease is really has an inflammatory features of diseases that are related, but severe inflammatory diseases like HLH. And so these will also be important in considering treatment options. What do we need to block, and what do we need to block at each stage of the disease in order to help these patients recover is a critical question right now. So with respect to this part, I want to just conclude that, again, this is extremely preliminary, but hopefully within a week or so, we would have a much better idea. But we are seeing some lower white blood cell count and monocyte count in the patients with progressive disease, and lower killer CD8 T cell features and higher TNF expressing Th1 cells in patients with progressive disease. And soluble factors like IL-6 and soluble IL-2 receptor appear to be elevated in patients with progressive disease, and higher levels of monocytes and dendritic cells are found in patients who end up recovering from the disease. So there's a lot more insights that are needed to understand the immunology to this virus, but we're making some progress here. So in the future, we would really like to expand these types of analyses to serial PBMC collected from each patient along the path, sort of natural history of infection for each patient. What happens when immune response goes awry, and what can we do to prevent and protect these patients? And serologic analysis will be incredibly important in parallel, because we're only looking at one aspect of the immune response, which is the cellular immune response. But clearly, the antibody will play a key role in both protection and pathology. So Aaron Ring is currently doing ELISA from these patients. So we will find out hopefully very soon what the antibodies' roles are in disease. And I just want to reiterate that this biorepository will serve the entire Yale community and beyond. And it's a really a testament to the leadership of Dr. Oliver Coe in setting this up, and so that everyone can benefit from these critical samples that are being collected. Okay, so since this is a cancer center grand round, I wanted to spend a few minutes talking about COVID-19 in cancer patients. And there are some issues that are probably very important to consider that is not applicable to other types of patients. And so I'm going to talk about a couple of reports that are out there. It's still very, very preliminary at this point, but maybe extremely important to this group. So this particular report came out in the Lancet Oncology, where it's a study of cancer patients with COVID infection, a national analysis that was done in China. And they did a prospective cohort study of 2007 cases of COVID-19 disease from 575 hospitals in 31 different regions. I mean, it's a very large survey across China. And they found that about 1% of the 1590 patients with COVID-19 had a history of cancer. And this was clearly higher than the incidence of cancer in the overall Chinese population. So the COVID-19 cases, patients are enriched for cancer. And lung cancer was the most frequent type that they saw within this 18 patients that had clear certified record of COVID-19 and cancer history. And so 4 out of the 16 patients with cancer with COVID-19 had received chemotherapy or surgery within the past month. So these are pretty recent treatments. And the others were cancer survivors and were kind of just being looked at in a routine follow-up. So there is a lot more data in this paper, lots of supplementary tables and so on that may be useful to this group. I'm not going to go into each detail, but here is a link to that paper if anyone's interested. There are a couple of sort of figures from this paper that is worth going over. So this panel here shows the incidence of severe events that happened in either non-cancer population versus cancer survivor versus patients with active cancer. And you can see that the severe events that were seen among these populations was much higher in the patients with cancer compared to those without cancer, or even cancer survivors have a much worse outcome in terms of COVID-19 disease. And so this is a kind of a reminder to just really we have to protect the cancer patients, because they're probably going to be suffering from more severe events if they were to catch this disease. And then this is a risk of developing more severe cases, severe events as characterized in this graph. And so patients with cancer have a hazard ratio of 3.56 more than the patients without cancer. And so again, this really it speaks volume about the need to protect the cancer patients from being exposed to this virus, as well as treatment of cancer patients that do end up becoming exposed and infected with the COVID-2. And there's another paper that was published more recently. This is also looking at transmission of COVID-2 in patients with cancer at a hospital in Wuhan, where it was the epicenter of outbreak in China. And so this paper, again, looked at medical records of 1,524 patients with cancer who are admitted to this particular hospital in Wuhan, cancer hospital. And they basically also found that the infection rate of COVID-2 in patients with cancer was higher than the incidence of all diagnosed COVID cases in the community at the same time period. Indicating that these cancer patients are likely more susceptible to acquiring this infection. And so the patients with cancer from the epicenter harbored a higher risk of COVID-2 infection, odds ratio of 2.3 compared with the community. So these papers are slowly starting to come out, revealing the importance of kind of looking at different types of approaches that may be needed for cancer patient treatment with this COVID-19 disease. And the immunology of cancer patients against the COVID-19 is something that I think this group would be able to really nail down and really look at in careful detail, because that's something that's going to be incredibly important to parse out for not just our community, but for community at large. And so here are some of my personal comments about special consideration for COVID-19 in cancer patients. First and foremost, cancer patients are immunosuppressed. So this status of even having cancer without any treatment, we need to be very vigilant about their baseline immunology and what that does to their immune response to the virus. The other ones are obviously the use of immune checkpoint inhibitors, whether that's beneficial or harmful or neutral to fighting this disease. It's unknown. That's something that I know that Yale New Haven Hospital has a lot of patients who are already on these immune checkpoint inhibitors, which we'll be able to kind of find out in real time. Use of rituximab or other agents that would deplete B cells or other populations in certain cancer patients would also be informative. B cells are it's unknown what the role of B cells are in this disease. Obviously, B cells are needed for the antibody to neutralize the virus, but B cells also make antibodies that are non-neutralizing, and that could even exacerbate the disease by activating macrophages through Fc receptor-dependent mechanisms. So what the role of B cells are would be revealed by studying patients who are on rituximab versus those that aren't. And then obviously use of chemotherapy and radiotherapy, and what that does to the immune system and what the impact will be for antiviral immunity, and surgery and many other treatments that these cancer patients are getting, and how that impacts on the resistance and disease tolerance against this infection would be really important to figure out. So I want to end here with by first acknowledging this amazing lab task force that we began this entire thing. This was early March that we still didn't have social distancing measures. It was a few days after this date that we decided that there shouldn't be any in-person meetings like this anymore. But this is just to, again, acknowledge the people involved in the biorepository work as well as other aspects. So Dr. Albert Koh from Yale School of Public Health, Nathan Grubel from also Yale School of Public Health, he's collecting RNA, doing genomic sequencing of RNA from COVID-19 patients and really trying to figure out the viral transmission behavior. So he's able to pinpoint where the virus came from, how it got there, as really interesting work that's going on. Dr. Sarah Omer from the Yale Institute for Global Health has been there from the very beginning, kind of coalescing these types of groups and trying to mobilize the entire community against the COVID-19. Ellen Foxman and Marie Landry are key members of this task force. Obviously, Dr. Marie Landry's lab is really kind of phenomenal job of making sure that patient samples are analyzed for COVID-19 RNA. And Ellen has been really spearheading this entire group, making sure that we have reagents and a protocol to be able to test for COVID-19 in the biorepository specimens. And so I'm very grateful to be able to work with these amazing people from the very beginning. And then finally, but I really want to thank all my lab members who have really stepped up to the task of everything that I talked about today. And these lab members, obviously, they have stopped all of their research that they were doing before and really devoting to PBMC collection, plasma collection, biorepository work, PCR, and their own research on COVID-19. So I am so thankful to be able to work with the brilliant individuals in my laboratory. Thank you so much for your attention. I'd be happy to take questions.
Akiko, thank you. That was fabulous. Great review of the work going on. I know you'll probably be able to, hopefully you can see on the bottom of your screen the questions and people are submitting questions both through the Q&A function and the chat function. But before we get to the ones online, I wondered if I could start. You obviously reviewed the role of the ACE2 receptor in the pathogenesis. And a question I'm sure you've been asked before and considered is ACE inhibitors have been really a revolutionary aspect of medicine in terms of hypertension, cardiovascular disease, diabetes. They're ubiquitous and as well as related compounds. Do you think that given the litany of individuals who are currently on those types of agents, that would have any influence on the natural history of this virus?
Yeah, so the ACE inhibitors, it depends on what the inhibitor does to the cells. If it's actually downregulating the receptor itself, that may prevent entry of this virus. But if it's interfering with the function of ACE2 without downregulating the receptor, it still allows the cells to become infected. And so there are lots of sort of, unintended consequence of using ACE inhibitor, depending on what it does to the surface expression as well as its functional inhibition. So those studies are, I know that currently being done by other groups to look at each ACE inhibitor, what it does to the viral entry process.
Can you see the questions that are being submitted?
Yes. Okay, Bram. How come you have neutrophils? Oh yeah, so there is a little bit of neutrophils that come in even though we do careful collection of the vical layer. So that's why we have this sort of extra neutrophil in our samples, but it's very small compared to what the neutrophil content in a whole blood, obviously. Next, Ezekiel. What are
I think
your
that was my
Oh,
question.
that okay, sorry.
Yeah, so if
Are
you can,
there there's Q&A. I didn't see Q&A. Okay.
yeah. And you
Okay.
may want to just read the question
Oh,
for,
sure. Okay.
yeah.
Jeremy J. Cox, Akiko, how do these cytokine profiles compare to normal? Oh yeah, normal healthy controls. So the ones that I showed you are all from the inpatients who have COVID-19. So we didn't have any healthy controls to compare to, but now we have the healthcare workers who are not infected with COVID-19. So we can compare these profiles head to head. And clearly in healthy people, we barely see any levels of these cytokines. And the other thing that I want to point out is that the more we test with respect to cytokines and cellular composition, we have a really robust group of the healthcare workers now that serves as a very nice control to all the things that I showed you today. So next time I give a talk like this, hopefully I can show that to you side by side. Anonymous, when do you foresee antibody testing to be available to healthcare workers, public? Yeah, so this is an important question because antibodies tell us a lot about our exposure to the virus and potentially immune protection. But antibody levels alone is difficult to equate to immune protection. But at least you know that somebody was exposed to the virus. And I know that the lab medicine team is currently already validating some of these antibody testing platforms for the hospital as well as potentially healthcare workers. For the public, I know that there are, FDA has approved several of these kits that you can use at home to test for COVID-19 antibody. I don't know the details about whether they're available and where you can get them, but I know that these things are coming online soon. Okay.
You might want to scroll down a little bit. Yeah.
Okay. Oh, there are questions that are very, very long.
Yeah, so you may want to scroll down to some of
Okay,
the
okay,
ones.
okay. Rates of increased COVID in cancer patients compared to general population are flawed in the literature because they are not corrected for age. Yeah, so that's why I suggested that you go into the supplemental data because obviously age is a huge factor in this. And so if you should look at the age group and looking at the cancer patients within each age group to be able to do comparisons. I just gave you an overview of what was reported, but I urge you to go into the literature to look at the other factors that are important. Next, two quick questions. One, is there any evidence suggesting any association between smoking and severe disease progression? Two, in your opinion, and based on how other coronaviruses work, is it possible to be reinfected with COVID-2 twice? What is known about immunological memory? Yeah, these are all very important questions. The first one, any evidence of association between smoking and severe disease. I think there was a medRxiv paper that looked at this very recently. I don't recall exactly the authors or anything like that, but I can dig this up. I think there is some correlation between smoking status and disease progression, in my opinion. Okay. Possible to get reinfected. Yeah. So there are reports, several reports showing that a person who was admitted to the hospital with COVID-2 and was discharged because of the PCR negative status came back to the hospital becoming COVID-2 positive again, PCR positive again. So whether that represents reinfection or whether that is a activation of virus reservoir that was hiding deep in the lung, because all of these tests are done with nasopharyngeal swabs, and that only detects the virus in the nose area, as opposed to reservoir that could be harbored in the lower respiratory tract. So that is really unknown, but there are definitely several reports of so-called reinfection or reigniting of the virus infection. So it's really unknown whether that's possible or not. With the common cold version of the coronavirus, the antibody levels do develop and people do become immune, at least for up to a year. Now, these immune response don't have a very long-lasting memory for the common cold version of the coronavirus. So it's unclear how long that immune memory lasts, and whether that develops in every person who recovers from COVID-2 is also unknown. And so this is really an important area of research. What is a correlate of protective immunity? Because once we know that, we can basically allow those people to come back to, to work and reopen some of the social distancing measures. But right now, we don't have a great answer to that question. Next, with the repositories, we are looking at creating a registry of cancer patients who have been diagnosed with COVID, who are on chemotherapy or immunotherapy. Are we looking? Yeah, what is your Twitter handle? My Twitter handle is Viruses Immunity. You can follow me, please. If Yale participating or looking to participate in the COVID-19 convalescent plasma treatment. Yeah, so that's something. The convalescent plasma treatment, this is not an area of my expertise, and I'm not sure if it's happening here at Yale. I'm not sure. With the repository, yeah, so Albert Coe and myself have been discussing this with Roy Herbst and others at the Yale Cancer Center to try to create a repository for the cancer patients here. And that would obviously have information about chemotherapy as well as immunotherapy or any other therapies that patients are on. So this is an important ongoing area of collaboration with the Yale Cancer Center. Okay, Stuart, is there a theory of cause of lymphopenia, viral effect on T cells? Lymphocytes are going deep into tissue such as lung. Yeah, so the lymphopenia only reflects the loss of lymphocytes in the blood. So obviously, it doesn't tell us anything about where the T cells are or whether they're dying. If you look at the respiratory tract T cell number, there have been some published in the medRxiv, shows that they're not particularly increasing in tissues either. So this means that lymphocytes are maybe not just going deep into the tissue, but they are actually being lost. So unfortunately, we don't have a lot of data on the respiratory tissue T cell or B cell phenotype. And this is something that the biorepository is currently working with Naftali Kaminsky and Charles Dela Cruz to be able to collect deep respiratory tissue samples from the patients so we can look at these issues. The theory of cause of lymphopenia, it is unlikely that the T cells themselves are directly being infected by the virus, and that's the cause of loss of the lymphocytes. So one hypothesis is that we're seeing loss of dendritic cells, at least from the peripheral blood, and that might correlate with the loss of T cells because there are no cells to be activating these cells. Or anyway, this is really early days. It's unclear right now.
Akiko, I can scroll down and read you some of the questions if that'll help. So I'll offer some. So here's a question: to what degree do you believe the central pathology of COVID-19 is a failure of host defense or a failure of host tolerance with attendant pathology? And what are the implications of this on approach to treatment?
Yeah, that's a very important question. And so if you look at the literature, and this is also consistent with what we're finding, is that the very severe cases correlate with cytokine storm type of features, such as elevation of many of these pro-inflammatory cytokines, as well as macrophage activation within the lung. So in the healthy respiratory tract, what you have is alveolar macrophages that usually capture pathogens and, destroy them and not cause any pro-inflammatory conditions. In the case of COVID-19 infected and severe cases, we see a loss of these alveolar macrophages and infiltration of inflammatory macrophages, which are capable of likely triggering this kind of cytokine storm within the lung. So I would say that it's likely the overt immune response to the virus that is causing severe cases of the disease. And so, yes, loss of disease tolerance may be at the core of the disease. And so that's why therapeutic approaches to boost host disease tolerance mechanisms or block the consequence of toxic cytokine storm, these are being tested, I think, right now. So in other words, I'm sorry, just to add to that, I think it's important to reiterate, because in the late stage of disease, antiviral measures are probably not going to be the most appropriate type of treatment, because by then the disease is already driven by the immune system. Whereas in the early phase of the disease, antivirals may still be effective in reducing the viral burden, because at that point we don't have this sort of cytokine storm yet. So hopefully that clarifies. Thank you.
That's great. So here's a comment that you may want to respond to. So the comment is, one thing that may be relevant in individual patient immune response is reactivation of other chronic viruses, especially HSV1 and HSV2, which tend to reactivate during stress. So thoughts about that?
Right. So the HSV1 and HSV2, which is really what I used to study before this outbreak, they tend to be very localized in either the oral or the genital tract mucosa, and its sort of dorsal root ganglia, where the latent virus resides. And so it's really unlikely to be found in the respiratory tract. So the reactivation of these other types of neurotropic viruses, may or may not have any consequence for this particular virus. But stress, on the other hand, in general, would have an impact on the immune resistance to the virus. So I think stress is definitely one of the key factors that we need to watch out for.
Yeah. So a question from Dan DeMeo: is expression of human ACE2 in mice sufficient to confer susceptibility to CoV-2, or might there be additional intracellular blocks for viral replication in mouse cells?
Yeah, so I think in vitro analyses show that kind of overexpression of human ACE2 in mouse cells alone is able to make them susceptible to infection. But in vivo, it remains unclear whether ACE2 alone is sufficient. However, ACE2 transgenic mice have been used to study SARS-CoV-1, which also uses the same receptor. So we're hopeful that expression of the ACE2 alone is sufficient to convert a mouse to be susceptible. But that's why I think I'm very excited about this collaboration with Richard Flavel's group to look at the humanized mouse, because in that mouse, you not only have ACE2 in the lung that we can introduce with the vector, but we also have an intact human immune system for which, whatever ACE2 expressing cell types are going to be there. So hopefully with that mouse model, we can recapitulate human infection even better.
Thank you. Here's an interesting question I hadn't thought of. It says there have been reports of BCG vaccine conferring immunity against COVID. Can you comment on this?
Yeah, so that's a very interesting hypothesis. I just wrote a commentary about that, so I'd be happy to share that when it's online. But yes, there is an association with BCG immunization status and very low level of death rate from COVID-19. And that's looking at countries like Japan and China and other Asian countries that still receive BCG vaccine as a childhood vaccine, whereas countries like the US have stopped doing BCG vaccine as a mandatory childhood vaccine. And some European countries that have high rates of death from COVID-19 also don't have a mandatory BCG vaccine requirement. Now, that's not to say that the BCG vaccine is conferring protection. It's just a pure association at this point. But there are trials of BCG vaccination happening in Netherlands of 1,000 healthcare workers. This is done by Dr. Nitia's group, who have been promoting this idea of trained immunity. Trained immunity describes innate resistance elevated by stimulation of the innate immune response. So regardless of the antigen, if you stimulate the innate immune response using something like BCG, which is a live attenuated vaccine, can one elevate innate resistance to other pathogens? And so that's sort of the idea behind this trial. So we'll wait and see what happens, but that is why there's this talk about BCG being potentially protective against this virus.
Thank you. Here's a question. Do we know how many variants of the virus have been recorded and whether they are distinct in terms of the severity of the disease?
Yeah, so Dr. Grubal would be much better equipped to answer this question, but he's been looking at lots and lots of viral sequences throughout the world. And I believe his opinion is that none of these mutants, so-called mutants, have any difference in terms of virulence or sort of transmissibility. But, I defer that to some expert like Dr. Grubal.
And does anyone at Yale currently study murine coronavirus, and to what extent is a murine coronavirus a good model for human COVID?
Yeah, so I believe that people here at Yale are studying murine coronavirus. And the beauty of studying murine coronavirus in mice is that it's a natural host for the coronavirus. And there are lots of features that are shared between human coronavirus and mouse coronavirus in terms of replication and disease. So that's another really great way to look at the immune response to coronaviruses in general. And I think at this point, we really need to have a multi-pronged approach. So that I think that's a really good thing to do.
Question is, what is known about the relationship between circulating antibody type and titer in antibody in the nasopharyngeal mucosa?
Yes, that's a really good question, because the serum antibody titer only can tell us about what's circulating and not what's in the lumen of the respiratory tract where the antibodies are needed for blocking the virus. So right now, I don't know of a lot of great evidence that shows the relationship between the circulating level of anti-COVID antibody versus the nasal or even the lower respiratory levels. However, the lining of the endothelial cells and the airway cells in the very lower respiratory tract, such as in the alveoli, are quite close to each other for good reason. I mean, for oxygen exchange and so on. And the axis of IgG is much more readily available at those sites than, let's say, upper nasal tract, where there are much more protective layers of epithelia. And the relationship between the blood vessel and the epithelia are much more distant. So I think looking at the IgG axis in the lower respiratory tract, as well as IgA, which is a much more sort of potent mucosal antibody that can be actually transported through a transporter from the lamina propria to the lumen of the respiratory tract. These are all really important things to measure within the lumen of the lung. And that's the other thing that hopefully Dr. Kaminsky and De La Cruz will be able to get at with their respiratory aspirates.
Well, Akiko, we know we're at the end of the hour, and we had a lot more questions. I'm sorry we didn't get to all of them. But thank you. This was a wonderful way to restart Cancer Center Grand Rounds, inspiring work to address this pandemic. Appreciate